only ever in detached leaf assays for cereal rusts and Phacopsera pachyrizae, which is soybean rust. And we wanted to kind of get into it a little bit more, really find out what was happening on the leaf surface and understand RNAi and rust fungi a bit better. So we really wanted to know whether rust RNAi was going to be amenable against all rusts in the Puccineales, so whether the RNAi machinery was present. We wanted to look, so rusts have complicated life cycles. We wanted to look which spore stages we could target with double-stranded RNA to trigger that RNAi pathway. We wanted to look at where RNAi acted when it was on a leaf and what it did to the rust as it was trying to penetrate a leaf in an infection. And then a big mistake of mine is that I was part of the molecular barcoding community. And so when I think about genes to target, I always just land on a housekeeping gene that we sequence for species identification. And it was later told to me that you shouldn't use housekeeping genes in RNAi because they're highly expressed and it's harder to knock them down. So this kind of became one of Beck's questions in her honors based on a horrible, unresearched mistake. We're going to get into it. So that first question of where the RNAi machinery is conserved across rust fungi, we looked at all these genomes of rust fungi. We sequenced genomes from Oricaria mysotaceae, which is a really rad genus on, a rad family on Oricaria. So super interesting taxonomically. The Sciurcaceae, Coleosporiaceae. We sequenced new genomes in Phragmidiaceae, Sphaerophragmiaceae, and Puccineaceae. And there's already publicly available genomes from Melampsaraceae and Puccineaceae. And past work on Phacopsera pachyrizae is in the Phacopseraceae. So we're kind of looking at 9 families, but at genomes from 8 families. And the message from this is that that RNAi machinery is present in every single rust that we looked at. So that's the kind of take home from this. It's present in every family across the Puccineales that we looked at. What's also interesting about this is that even though we've got paralogs and orthologs of our RDRPs, our dices and argonauts, they're just homologs of the genes that were originally identified in Ascomycota and Cryptococcus for these RNAi machinery genes. So since rust shared a most recent common ancestor, those genes have gone their own way and they're not really identifiable as orthologs of what's in the Ascomycota, but they're still there as RNAi machinery genes or homologs, gene homologs. So we knew that rusts had the machinery. We then needed to take that to the streets or to the leaves. And this is for a bit of South African nostalgia. This is Ostroppuccinia sidii or myrtle rust. And I photographed this in the Wilde Coast, probably in 2017 or 2016. And these first 2 images are of 2 endangered species of Eugenia in the Wilde Coast. I would love to know what's happening in those populations since then, because when I looked at them, they were already starting to be matchsticks. But Myrtaceae is such a small part of the environment of South Africa, unless you're standing in a eucalyptus plantation, compared to Australia, where kind of our ecosystems are just a giant eucalyptus plantation. Depending on what ecosystem type you're in, you're probably looking at a Myrtaceae. You got a 50-50 shot. So it was always known that myrtle rust was going to have more impact in Australia than in South Africa, where it's exotic in both countries. So visually, when you want to visualize this impact, this is a secondary growth rainforest near where Louise and I live in Brisbane. Some Fabians have been lucky enough to see it. This is supposed to be a rainforest, and you shouldn't be able to see the canopy through this forest. So, sorry, you shouldn't be able to see the sky through the canopy of this forest. So everything you see here is pretty much a dead Myrtaceae. When you get up into that little kloof, that little hill, Louise, Jeff Pegg and I, we were doing these little plots. So a 10-meter radius in a circular plot, counting about 110 trees. 2 dots means a dead Myrtaceae. 1 dot meant a living one. And we were counting 90 to 100 dead Myrtaceae per plot all through that site. So it would be great to have a way to manage myrtle rust in Australia You can't just go into this natural ecosystem and start spraying around fungicides without kind of that Agent Orange scorched earth effect. New Zealand also would really benefit from being able to protect sacred trees from myrtle rust, where they don't want to spray fungicides on sacred trees. So RNAi is a pretty good application against myrtle rust if we can get it working. So we looked also at, that was all just myrtle rust. It was an introduction to a rust. Whew, gosh, hopefully you're on the same page. Then there's this other rust, and another piece of nostalgia. I photographed this in South Africa. In Brisbane, this is beside every pool. It's in every backyard that has a frangipani. It's called Coleosporium plumeria leaf rust. And these two rusts were just our models that we were testing RNAi on. So both of these rusts, they're different to cereal rusts. Cereal rusts form spermagonia and acea on an alternate host a lot of the time, or other species of Puccinia might form spermagonia and acea on the same host. These two rusts both just have a uridinial stage, which is a stage that just spreads the same genotype as a clone. And then when in a basidiomycete or a basidiomycota, most of their life cycle is spent as a dikaryon with two nuclei from their basidiospore parents. Teleospores are the site where those nuclei fuse and you can have meiosis. Teleospores germinate to produce a phragmobasidium that produces recombinant basidiospores after meiosis. So we're looking just in teleospores and uridiniospores. And in a normal rust life cycle, a uridiniospore will land on a leaf and it's got these little germ pores, and from those germ pores sprout a germ tube. So one will often anchor the spore, kind of like, hey, I've got a hold of this. And then the other invests all of the cytoplasm from the cell and pushes it down through cytoplasmic streaming into the end of the apical growth of that germ tube to form an aprosorium. And that aprosorium, with all of the pressure from the cytoplasm, builds up enough force to penetrate through a stomata or directly through a leaf cuticle with a penetration peg. And in my 10 years of working on rust fungi, I had never seen one. Oh my gosh, you guys are in for a treat soon. Okay, so we were looking at uridiniospores and teleospores. The first experiment that my colleague Anne Sawyer was doing, and you're going to see photos of all of this. I just want to prepare you for what you're about to see with a simple to understand cartoon. So we tested double-stranded RNA uptake, which is kind of the trigger to trigger RNAi in rust fungi. We looked at that uptake in uridiniospores and teleospores. So what you'll see is that uridiniospores take it up, but teleospores do not take it up. And teleospores are just germinating not to cause an infection, they're germinating to produce that phragmobasidium that has the gametic spore stage. So what you'll also see is when we apply double-stranded RNA that targets a rust gene, is you'll see that the germ tubes themselves start to have some changes. If the spore germinates at all, so the germ tubes are really stunted, or they'll be super withered and kind of, you feel sorry for them sometimes. They never get to form an aprosorium and they just look kind of pathetic. So keep your eyes peeled for seeing some of that in the flesh in just a second. And so what made this all possible for Beck's experiments is our colleague in New Zealand, Grant Smith, who wanted to test these silicone leaf discs to extract RNA in the plant pathogen interaction. But Grant was sick of discarding 99.9999% of plant reads in his transcriptomic studies and using, small numbers of reads to do all this transcriptomics. So he wanted to cut out that middleman that was the leaf. And so he was really keen to try these silicone discs, which have been used since the 50s to trick rusts into thinking they're on a leaf. So Louise was kind of like, okay, Louise Shuey that is, was like, well, let's take it a step further and test and get Beck to test some of her double-stranded RNA work on this. So Beck would mix up her little spore suspension solution and then apply that with double-stranded RNA onto these leaf discs. So this is where I was like a caveman being introduced to fire, because I'd only ever read about aprosoria and never gotten to see one. And so this is that classic, all that cellular contents has come out of the germ tube, been invested in making that aprosorium and then forming that that penetration peg. And yeah, I think not many people get to see them. So these silicone discs are just so unreal to be able to study rust physiology. What we also got to see is this cytoplasmic streaming. So just before aprosorium formation, and then you'll see that the aprosoria here have they've pushed out all their cytoplasm and invested in that penetration peg, getting through whatever they think is a leaf cuticle or stomata. So this is what Anne did to test uptake of double-stranded RNA in different spores of rust fungi. So uridinospores of myrtle rust and uridinospores of Coleosporium plumeriae. And when they germinate, they take up double-stranded RNA, which is labeled here with Cy3 so that we can see the uptake. The teleospores, you'll notice, are not glowing orange, and that's because they didn't take up any double-stranded RNA. So these structures aren't anything that can infect a host. As I said, they're just phragmobasidia. So what's interesting is that if you were trying to stop meiosis and production of basidiospores with RNAi, I don't think you could. But a future question would be whether basidiospores take up double-stranded RNA when they germinate. So we know now that uridinospores take it up, but teleospores do not. So when we physically target a housekeeping gene like beta-tubulin or TEF, we end up with this kind of long, winding, poor germ tube that it's just so unsatisfied. This one, the TEF one, you can see it just looks withered and it's never going to form an aprosorium, the poor things. So something's going on with the physiology when you start to knock down these housekeeping genes. And what Beck found when she did her spore germination assays was a significant reduction in germination rate of myrtle rust and a significant reduction, even though it already had a really low germination rate, in frangipani rust, especially with LSU. So Beck was also just checking whether double-stranded RNA impacted either the aprosorium formation or germination rate or whatever, just as another positive control. So this was nothing, and this is just kind of with double-stranded RNA labeled with GFP, just to check if that double-stranded RNA was having an effect. So these are just that with labeled double-stranded RNA labeled with GFP. So there's those infection pegs, aprosoria, Coleosporium plumeriae looks a little bit different. But then you can see when we start to knock down with RNAi with these housekeeping genes, here's beta-tubulin, just pathetic kind of germ tubes. Here's that interruption, clear interruption of cytoplasmic streaming. And then with LSU, it's even hard to find spores that have germinated. And this is kind of the best effort at a rust spore making an aprosorium when it's been knocked down with a housekeeping gene. So this is probably the first use of artificial leaves to look at RNAi in rust fungi, probably with all fungi. So that's pretty neat. So Beck knew that it could cause an effect at the leaf surface, and she took that to the leaves and she did some inoculation studies on Syzygium jambos, and very similar experiments to our artificial leaf experiments. So with no double-stranded RNA, with GFP labeled double-stranded RNA, which still caused an infection, and then significant reductions in infection rate with these housekeeping genes, beta-tubulin, to the extent that it couldn't even cause an infection. And Beck is the first person to ever do a whole plant assay with double-stranded RNA for rust fungi, which is pretty neat. Nice work, Beck. So the outcome of all of that is that we know that the RNAi pathway is present in these families of rust fungi, in 9 families, including Phacops raceae. And we think it's probably amenable to control rusts using RNAi all across the order, if you have a rust problem in agriculture. We know that uridinospores take up the double-stranded RNA, but teleospores do not. We haven't tested other spore stages. But this is good news for if you do have a clonal outbreak or an epidemic caused by uridinospores, then double-stranded RNA or RNAi as a disease management strategy could be used. We know now with that innovative use of artificial leaves that RNAi stops the aprosorium formation and translates implanter to reduce symptoms. So all pretty cool. And then that last question was whether we could use essential genes as targets for RNAi. And my colleague Anne reckons that we can, that it's so effective in rusts for a hypothesis that is to be tested because of RDRPs, which might be active. So the cool thing about these essential genes is that they really can just be pulled off the shelf. You don't need to know the sequence of the rust. You can just use universal primers, get some DNA of your rust, to synthesize double-stranded RNA, and you're ready to go. If a rust is going to break resistance to RNAi, then it would either need to mutate at several places along a housekeeping gene, which is easier said than done. That's just because after the double-stranded RNA is cleaved, there's so many different places that it can be used to knock down the transcripts. Or if it also wanted to break resistance, it would have to maybe have a massive mutation in that RNAi pathway, which also is probably easier said than done. Okay, so that's the rust part over, onto the sex. And what you see here is, well, I'm just going to give you a second to digest it. If you're a plant pathologist, you might recognize these as former speciales of fusarium oxysporum. And so in that fusarium oxysporum community, if you're working on a plant, you'll bung a former speciales, whatever, on the end. Cubensi is this fusarium oxysporum on banana, but it's pretty unsatisfying for taxonomists when you reconstruct a tree and you've got cubensi in all sorts of crazy locations and they don't form a monophyletic group. So in lockdown, in COVID, we were kind of watching this taxonomic debate going on in fusarium, which is always super entertaining to watch one of these. And you've got one camp that say that there's a lot of biodiversity and then another that say that there's 1 to 3 species. And, you know, I don't have any skin in the game. I'm pretty indifferent. I still find it really fun to watch, though. The outcome of all of this is that there's a huge amount of data for a data parasite like me to take off GenBank. When I downloaded these data in, I think, October 2020, there were 500 genomes of fusarium oxysporum just sitting there waiting for the plucking, waiting for something other than a taxonomic question. And I always thought that that taxonomic question would be better asked from a sexual reproduction kind of angle. And with Tuan and Brenda and some of our other colleagues, all who were kind of separate to the fusarium community. Brenda had dabbled in fusarium, obviously, and Brett Summerall was also involved, and he had some skin in the game, but everyone else was totally oblivious to what fusarium did, including me. We had no idea. We took the single copy orthologs from about 410 genomes, and we looked for structure, population structure a priori. So we just pretty much said, okay, well, these are the populations. Now let's look in these populations for these things that are evidence of sexual reproduction, like segregated mating types or lots of non-concordant topologies within a phylogroup, or random inheritance across a massive alignment of genes and reticulation in networks. Similar to looking at genes, we looked at SNPs. We looked at about 4.2 million SNPs, rejected linkage disequilibrium in all those phylogroups just to say, okay, well, you've got SNPs inherited randomly. And then RIP, which everyone at FABI is familiar with. Stefani, loving watching what you're doing. Can't wait to see what you do next. Tuan did the RIP work. Unfortunately, we didn't get to use Stefani's pipeline, but we showed that RIP was present in every single phylogroup as well. So all strong evidence that there was sexual reproduction in the past, and what we think is evidence of sexual reproduction ongoing in these different phylogroups of fusarium oxysporum. And, you know, a braver man than me would say that those phylogroups could be thought of as species because there's clear boundaries of sexual reproduction. And what probably the take-home message from our work is that there are these phylogroups where there's recombination within a phylogroup, but not among them or not between them. So that's kind of the big thing, which is a good boundary for a species if you were invested in the taxonomy. But when we were getting this reviewed, there was a big, hey, wait a second. You guys have only looked at the core. We looked at single copy orthologs And then to call SNPs for our SNP study, of which there was 4.2 million, we only called a SNP if it was present if that locus was present in every single genome, every single one of those 410 genomes. So the reviewers said, hey, well, what if you're only looking at the core, but all the evidence of sexual reproduction is in the accessory genomes, or there's horizontal exchange of accessory genes or chromosomes, and you're only concentrating on sexual reproduction. And this is just a homogeneous group of one fusarium oxysporum, and you're trying to say that the boundaries of sexual reproduction are what is delimiting taxon recognition. So that was still looming over us afterwards, and that's what I want to talk about right now. That was all background. So yeah, this was keeping us awake at night. Obviously, lots of things do. So we just wanted to ask, is there evidence of recombination among those phylogroups? I've written species here. That is a Freudian slip or something. I don't know. That's an honest mistake. We wanted to look for if there was evidence of recombination among those phylogroups within the accessory genome. And then what was also super relevant was whether there was horizontal exchange of DNA among those different species, among those different phylogroups of Fusarium oxysporum S. lat. And it's really relevant because of this paper, which is probably my favorite paper in plant pathology ever, by Li Junma and colleagues. And if you're not familiar with it, well, you should be. And I'll just take you through it in about 30 seconds. Ma and others took a pathogenic strain of Fusarium oxysporum. It's pathogenic on tomato. There was a non-pathogenic strain. They aided the movement of a chromosome to the non-pathogenic strain, the dispensable chromosome, chromosome 14, the infamous chromosome, and that non-pathogenic strain became infectious on tomato. So imagine that. If that chromosome moves willy-nilly in Fusarium, on the whim of the breeze, of its own volition, then why do we even need these species names anyway? We should just be tracking that chromosome. So how often does horizontal gene transfer occur? Is it something so frequent that we should be worried? Or is it something that's rare in the evolution of these organisms over time? So we asked these questions with a much reduced data set. We used 136 genomes, which meant we could also look at more genes. So this is just the single copy orthologs again. This time we could analyze 850 of them. And there's nothing different between those single copy orthologs from the core genome to our previous study. So we recover the same phylogroups. So we can almost forget figure A now. So B and C are analyses of the accessory genes. And figure B, each of these colored circles is a genome colored by their phylogroup in figure A. Just said we could forget figure A. Well, now you can. And then each of these little gray dots is an orthologous cluster of genes. And the way that these networks are arranged is by their shared relationships of those orthologous clusters of genes. So when you have lots of color clustering together, they share lots of accessory genes. So I've just deleted all shared orthologs in the core. And I've also deleted genes that are present in every phylogroup, which could be a problem, as we'll discuss later. But yeah, based on this network where the colors all group together, I would lean towards saying that there's no evidence of exchange via recombination among the accessory genomes. And then just to visualize that again, this is a splits tree network based on just the accessory genes used in here. And so things are grouped together based on a presence-absence matrix of those accessory genes. And so we do recover our phylogroups essentially. If we were going to be certain of recombination among accessory genes, we kind of expect a little bit more connection up around here. This is pretty interesting itself, which I think is probably something to do with this genome here way down near phylogroup 5. But yeah, I would say that this is pretty clear that there's no sexual reproduction or recombination in that accessory genome, or certainly no evidence of it, no clear evidence. So it's still all happening within those phylogroups and not among them. So the horizontal gene transfer component is a little bit harder. So I think we all know that horizontal gene transfer, if we're using a phylogenetic species hypothesis and we're looking at a inter-domain transfer or inter-kingdom, if you've got a group of fungi that you think have inherited a bacterial gene and you look at that species tree and there's bacteria and fungi and monophyletic groups, and then you look at the gene tree that was horizontally transferred and suddenly your group of fungi is up with bacteria, then that's chalk and cheese for horizontal gene transfer in your gene tree and a species tree, which is how you expect those individuals to be related. Well, looking in closely related species is a little bit trickier because your phylogenetic hypothesis, if there's been recombination in their history, is going to be complicated. So we can't just look at single genes. We found that we had to look for long pieces of DNA, because we're still doing it based on genes, because it's still the easiest way. We had to find patterns of individuals that had the same genes and then go back and trace them back to where they sat on the genome to look if they were all in the same region. That's going to make sense when you see an image, I hope. Then the next trick for identifying horizontal gene transfer is to distinguish it from just natural gene loss. So is it definitely a gain from a horizontal event, or has it been that from a most recent common ancestor, every individual in that phylo group has since lost the gene except for one individual? And wouldn't it be embarrassing to say that that individual had inherited something horizontally when it was just the sole possessor of a gene that had been present in the most recent common ancestor? And then, this I'll cover in a little bit as well. So actually, because I stuffed up explaining how genes should be in the same area, I'm just going to go to the next image real quick. So we identified two kind of parts of the genome in 13 individuals that we thought might be evidence of horizontal gene transfer. And what's super interesting is that those two kind of regions were both in the dispensable chromosome, so chromosome 14. And we identified them by finding patterns of gene presence in these 13 taxa. These first, I don't know how many, these first couple are phylogroup 6 and the rest are phylogroup 5, 1, 2, and 3. So this is about 4 genes, and then the other one has 3 genes. So, and they're not present in any other taxon in our data set. So I hope maybe that cleared things up. I'm sorry I butchered it so badly. So then when we look at this network is just chromosome 14 accessory genes. So I've deleted all of the genes that are shared by every phylogroup, and this is only about 2,000 genes structuring the layout of the relationships of these genomes. And what you'll notice, and one more caveat, the way that we define chromosome 14 is based on phylogroup 6, which has the best assembly for chromosome 14. So phylogroup 6 is looking pretty tight, but that could be because we used it as the foundation of chromosome 14, or the dispensable chromosome. But yeah, what you'll see compared to that last network is that we don't have lots of these colors clustering together. The phylogroup 5, this green one, we've got it wedged up with phylogroup 6. What's the beef? We know that something's up, and maybe it's to do with these genes that I've highlighted in red that are those shared things on those 2 long 10 kb to 20 kb fragments on the dispensable chromosome. So we think that those are candidates of horizontal gene transfer based on their pattern and shared nature in really unrelated phylogroups by recombination. So the next stage is to show that those clusters of genes are a true acquisition rather than a massive loss from those phylogroups. And unfortunately, I've hit a little bit of a bottleneck, because the most parsimonious explanation to explain those potentially horizontally transferred genes is that they're lost in every single other of those 136 minus 13. They're all lost from the most recent common ancestor of every phylo group. So it's a little bit tricky to navigate showing that it's a true gain over a loss at the most recent common ancestor. So yeah, from what I've just shown you, I think it's evidence that accessory genes support our past finding that there's no recombination among different phylo groups. Probably horizontal gene transfer is infrequent, and that's based on how difficult it was to even find our candidates that are putatively horizontal gene transfer events. Of course, there's the alternative hypothesis that we've missed many events by deleting the genes that were present in every single phylo group. I may have deleted things that have been duplicated and then shared, but I probably definitely did. And also by decreasing our data set to 136. Maybe I've missed the cream. So yeah, I'm kind of leaning towards horizontal gene transfer is infrequent though. The next step is for me to trawl through even more genes, essentially. And this is where my ulterior motive kicks in. If you are intrigued by any of this, then you could come and work with Tuan, Don Gardner, Tim James, Brenda, and even me, as annoying as it could be. And we'd love to hear from you if you want to help. Those data are all publicly available, and I'll just share with you where we're up to. If you can, get on board. I'm sure you've all got heaps of time. Okay. Last part. Oh, gosh, I'm running out of time. The magic. I'm going to have to zoom through this. I'm sorry, Liesl, for you joining in and only getting a tiny taste of magic mushrooms. I think of this not as my work, but as the work for everyone, just because there's such an interested community. If you want to see what I'm doing, anytime I do an analysis on magic mushrooms, I just pop it on the old bloggy. Now, if you don't know what I'm talking about by magic mushrooms, you may have been selected as an Elon Musk participant and have been living on Mars. Kudos. If you open up any newspaper at any random time of the month, there's probably a story in there about psilocybin and its breakthrough treatments and promise for mental health, PTSD, palliative care, all sorts of things. One of the most recent ones I read, and I can barely keep track of them all, in a study of depression, 1/3 of patients were in remission after one dose, 6 months after that dose, which is incredible when you think of the cocktails of drugs that people with depression have to take. So psilocybin is a real breakthrough promise, and it was where I wanted to start directing my research activity. And I'll just say, I think, humans are interested in humans as the main model organism, but we haven't looked at how we can innovate psilocybin in animal welfare. So I think there's so many gains to be made in the future. I just can't help doing this just to help you identify them in South Africa. So species of psilocybe are pretty easy to identify because, well, they're little brown mushrooms. A lot of the time they have a purpley brown spore print. You can see this Pileus has dropped a whole bunch of spores on the Pileus of this mushroom. And they've got gills. So when you make a spore print, you want to have gills there. The other little interesting thing is that species of psilocybe have 4 to 5 genes in a cluster that metabolizes tryptophan into psilocybin. And psilocybin will often stain blue when you damage the mushroom. I think it's an oxidation of psilocin. Technically, if you were using these mushrooms recreationally, which I certainly don't do because that would be a breach of my permit, you don't want to damage a mushroom too much. Now, in this mushroom up here, this is not a species of psilocybe. This is a species of Paniolus, and it is in the Agaricales, but it's not a related family to psilocybe. And it's thought that it inherited those 4 to 5 genes that produce psilocybin by horizontal gene transfer. That's in a really cool paper by Reynolds et al. In 2018, if you're interested. They share an ecological niche, and there are other competing hypotheses that maybe psilocybin is a convergent pathway, but this was quite strong evidence because that cluster of 5 genes is present identically in Paniolus. So the group that you need to watch out for is the sister genus to psilocybe. This group of little brown mushrooms is called Galerina. And instead of producing psilocybin as a secondary metabolite, they produce a metabolite that gives you organ failure in 10 to 24 hours. They produce amatoxins, and they grow in really similar environmental niches to psilocybe. They're really easily identified by their rusty brown spore print, and they would not stain blue if you ever picked them. Okay, that's all of that in a nutshell. And then I've just got heaps of photos of psilocybe that I like to share, but we're so short on time, I don't think I can. There's that little bluing reaction. And then this is a really good identifier of psilocybe cubensis, is this annulus, which stays there as a remnant of the veil as the pileus opens. So yeah. And then there's that purple spore print of psilocybe. And then these are black spore prints of paniolus. When I was studying mycology in the States, they kind of said, you can't die from eating a purple spored mushroom. I mean, that always comes with the caveat, though. There's exceptions in biology all the time. And wouldn't it be terrible to be that exception? So I guess you usually need to know what you're doing. And then there's those spores in the flesh. I kind of want to take you through this, but I'm mindful of the time. I'm so mindful that Fabian's ascomycete people to the core, except for the exception of Martin, bless you, and Brenda a little bit. So I'm going to take you through it. I just convinced myself. Basidiomycetes, they're dikaryotic in most of their life cycle. So they've inherited the nucleus from their two parental basidiospores. They maintain fidelity of that dikaryotic stage by these little clamp connections. What's super interesting is that rust fungi in their limited hyphae don't produce clamp connections. So rusts, it's unknown how that nuclear fidelity is maintained. So when you've got a mass of mycelium or hyphae or a fungal hymenium developed and you meet the right environmental conditions and that mushroom says, okay, it's time for sexual reproduction. In those gills, you produce a basidium. That basidium is the site of karyogamy, so the fusion of those two nuclei, and you have production of those 4 daughter basidiospores. The mating, which is not fully resolved, but which I intend to do tomorrow or on the weekend, it's comparable to what you guys know from what Marcus is doing. It's just that in Basidiomycota, you have tetrapolar mating, which means that mate compatibility can be controlled at 2 loci. So you must differ at those 2 loci to be compatible. And it's just a mechanism that stops inbreeding if your spores from your parent are landing in the same location. So you end up only being able to cross with one of your daughter basidiospores rather than 2. The haploid stages of Basidiomycota are always really pretty underwhelming, but they're the stage that I work on, obviously, for genetics. In smut fungi, they're non-pathogenic, they're the yeast phase. In mushrooms, sometimes you'll have a pathogenic haploid stage and it will form a really weak infection. But most of the time, they're just looking to get that extra copy of their chromosomes. And in rusts, that's the spermagonial stage, which, is forgettable. Okay, let me just check the time. All right, we're not going to have much time for questions. I'm going to flash through this. So this is the background of the research that I'm currently doing. So we've got 2 mushrooms that are prolific in Australia that produce psilocybin, our flying saucer mushroom, or in the underground psilocybin community, these would be called subs, Psilocybe subaeruginosa. What's super interesting about them, they were described in 1927 by a guy in South Australia. He described them from New South Wales, Victoria, South Australia, so in that southern east corner. And they really occur in Tasmania a lot as well. And I've collected them up in Queensland, and they also occur in WA. 20 years after that description, a very, very similar looking mushroom was described from the UK, from Kew Botanic Gardens, from leaf litter in a planted garden bed. It was described as Psilocybe cyanescens. And maybe, as people who are living in the Southern Hemisphere, you're thinking the way that I'm thinking, that well, in Australia, it's certainly true. We apply a lot of Northern Hemisphere names. The Northern Hemisphere is streets ahead in terms of knowledge of biodiversity of fungi. And it's really rare that something would be described here before it's described in the Northern Hemisphere. So psilocybe, not subaruginosa, but cyanescens, the one described from the Northern Hemisphere, has spread all over Europe, and it's spread through the west of the United States. I should also say that when these were described, psilocybin was unknown in Western science. It wasn't, psilocybin, the activity of it wasn't described until 1958 by Albert Hofmann, who is also the person who identified LSD, lysergic acid. But when this mushroom became widespread in Europe and America, it became a go-to recreational taxon. It started to get patented for the extraction of psilocybin. It catalyzed what we now have for the baseline of how psilocybin benefits mental health. And that is the trigger to my research question, which is I want to show that this mushroom is endemic to Australia, including psilocybe cyanescens, this Northern Hemisphere taxon that's been patented by people in Sweden and America and whatnot. The mushroom itself now is not patented, but at some point in time, the knowledge of the use of psilocybin under the Nagoya Protocol and also under the Queensland Biodiscovery Act, there should be some sort of flow-on benefit sharing agreement with people who originally discovered the use of psilocybin or the countries where these mushrooms originated. And as South Africans, I'm sure you can empathize with people coming to Africa and taking things elsewhere. So the other mushroom that we have prolifically in Australia is psilocybe cubensis. And I believe that this is an introduced taxon. It grows out of cow manure. Now, I'm going to cut this talk short, but this is where I want to plant a seed with you. This was originally described from Cuba, but it looks really similar to a mushroom that occurs in South Africa called psilocybe natalensis. And this mushroom is the other mushroom widely used for production of psilocybin. So it would be super interesting to find out its true center of origin. So really, I think I've kind of explained those questions. I would have taken you through what I do. I've assembled all these genomes from a couple of populations. My population sampling has gone up over Christmas while psilocybe cubensis has been fruiting here. I think I've got about 8 populations and more than 60 cultures. So I'm almost ready to kind of determine whether this is a true introduction. If it isn't an introduction, I'm going to have to rethink my sampling. These are such tiny genomes compared to rust fungi, only 50 meg. Brenda, when you introduced me, you spoke about the myrtle rust genome, 1.2 gig. Oricariomyces was 5 gig estimated from, what did we do? From chromium sequencing. So yeah, really, there's one other interesting thing here. This little genome is the Dikariotic assembly. So the number of contigs are in the center of these circles, which are proportional to genome size. And I was really worried I was sequencing dikaryons and my selection of haploids was atrocious, but it looks like it's all working out. So that's nice. I'll just quickly tell you about one more story just while we have time. There's all this soil sampling data in Australia where they've gone to remote locations in Australia, to national parks, and just done ITS2 barcoding across soil samples. And I was just looking at the distribution of psilocybe cubensis. So my hypothesis here was that this gold top was going to be all over the place because it's like a weed. It's like lantana growing across Australia. And amazingly, it wasn't. So it wasn't present in any of these publicly available data sampling sites from all across Australia. So what is present is psilocybe subaruginosa from all these national parks, and then a couple of other species. But this one really stood out because it's present in Kakadu National Park and Litchfield National Park in the Northern Territory. And this mushroom, whatever it is, we don't know, and psilocybe subaruginosa are definitely hallucinogenic species that have predated the arrival of humans 60 to 100,000 years ago. And one really interesting question that I guess I'll never be in a position to answer is whether Australian Aboriginal people were aware of the benefits of psilocybin, similar to Mesoamerican cultures. But yeah, that's not for me to answer. So really, I guess what you learned from my talk is that RNAi acts at the leaf surface. You saw it in the flesh, knocking down those aprosoria, withering those germ tubes and stopping infection. We think that that's going to be applicable to rust fungi across the order. Please help if you want to with showing horizontal gene transfer in fusarium oxysporum. We think that the accessory genes do not support reproduction among species. And then you can follow any of my magic mushroom work at the blog. And I reckon, gosh, imagine if Psilocybe cubensis is native or is the center of origin. The South Africa is the center of origin. That would just be so cool to know. Maybe one of you can do it. Thank you for listening.
Thank you so much, Alistair. I'm sorry you had to cut that short. I'm kind of thinking we need to invite you back. Lost for the game. But let me just start other than saying thank you very much. That was wonderful. You took us in 3 different places very, very rapidly. There are some questions that are being asked in the chat line. And the first question that we have here is from Albert Hamabaka. And Albert is asking about the silicon discs that you use for rust germination. So I think maybe she's asking where she can get them and yeah, how she can use them.
Hi, Almut. I reckon that your work would really benefit from those discs. I'll put you in touch with Grant Smith and Louise, who can help you out. They can really tailor it for anything. There's all sorts of applications, especially for leaf pathogens.
Great. Thanks very much, Alistair. I think that's answered Almut's question. Then Stephen, who's actually in the same time zone as you, he's down in Melbourne. Stephen routinely joins, well, Stephen's still part of the University of Victoria, but routinely joins us on Zoom. So Stephen is asking, he says, Alistair, to my knowledge, RNAi is highly conserved across eukaryotes. Are there many cases in the literature that reliably demonstrate the loss of RNAi machinery that demonstrate the loss of RNAi machinery in some fungi? It seems too important a defense and developmental adaptation to herbs.
Yeah, right on, Stephen. It's such an important pathway to control or just to regulate post-transcriptional expression, right? So it's unlikely that it would be hard for an organism to lose that pathway as developing resistance. In fungi, there's been various studies that show activity of RDRPs and whether you have that upregulation of the pathway itself.
And maybe I can just also add there are a couple of us in FABI who are actually looking at various aspects that involve double-stranded RNA. The one thing that immediately comes to mind is if you're looking at RDRPs, you might not actually be looking at fungal genomes, you might be looking at virus genomes. And that's another thing to think about, but you don't have to worry about that if you're looking at RNA data. But my understanding is that the double-stranded RNA has not worked on everything. And I think that's, maybe to answer Stephen's question, is that's one of the reasons certainly why I think the first thing one does is to look for all the genes that are involved in that pathway as a starting point. So the next question is from Carla, and she says, thank you very much for the seminar. And she's asking about what concentration of double-stranded RNA was applied in planter. Maybe you should just send us the publication.
Yeah, well, like I said, this is as fresh as a pick and pay shelf. Nothing here has ever been published. We're looking for a home for that piece. Carla, that is a lot of double-stranded RNA. I can't remember what Beck used, but it's a lot. It's like probably, I think it could be a microgram. So yeah, it's a lot. Actually, Louise is here. Do you know how much it was? double-stranded RNA applied in the experiments?
It's less than a microgram.
It's less than a microgram. Sorry. That's going to be a mystery now. You read about it in the...
Follow, one of the answers to the questions also is there is quite a bit of, Alistair's not the first one to apply double-stranded RNA in plant. There's quite a bit of other literature out there. So not trying to burst your balloon, Alistair, but that the rust stuff is really lovely. And I would have thought you would but anyway, you need to
It's the first rust work implanter, absolutely. All the other rust work is on detached leaf assays. So that's what I was saying. I'm sorry if that was misinterpreted. I'd feel terrible to all the RNAi community if they thought I was misrepresenting our work. Oh, man. I'm going to prostrate myself.
So Carla says thanks, she'll read about it. And then there's just a comment from Deborah when you were talking about the few people who are actually working with mycetes. And Deborah's a PhD student with us working with Martin and myself.
Oh, bless you, Deborah. Keep smiling. By that flag.
Oh, and then we've got the peanut gallery in the background here. Eriska's going to start working, or has already started working in Ganoderma. So
happy birthday.
And
Jan has a question. Jan, because it's a scheduled substance, it was a lot of effort to get a permit.
And Bernard says don't forget about the Alistair. Sorry. There is quite a bit of basidiomycete work done.
I'm a slanderous person, aren't I?
And then Jan is asking, it says, do you also culture psilocybin? I think he asked that question after you, before you actually talked about some of the work you were doing.
Okay.
His real question is, what are the legal issues related to working on these fungi in Australia?
Yeah, Jan, it's a bit of a headache just because it's a Schedule 9 substance here. It might be really high up in South Africa as well. So it's considered the same penalty as heroin for possession. Of course, everything that I work on, you can't grow a mushroom from because I'm just growing haploid stages. So I think they looked at my background and then I was in the right place to be doing it in a herbarium, and they gave me a permit after a bit of time. But they were scratching their heads because they kept saying, how much psilocybin are you going to be using? I'm like, none. Oh, I don't know. So, yeah.
So, but Alistair, what you haven't really explained to us, I know you rushed through some of the work on psilocybin, is what exactly is the project that you're doing? You touched on
well, the project's not funded. I'm really just testing endemicity of our two main species. And then based on, because I will sequence genomes of everything just because it's so easy and cheap, there's so much data there to then play with. So I'll be testing species boundaries, looking at cryptic species diversity in Psilocybe subaeruginosa, then looking at that cluster of 5 genes to look at diversity in the expression of psilocybin. I've hooked up with some chemists. We'll see if we can start looking at the phenotypic fingerprint of psilocybin across the population in Australia, and see if we can trace that back to variation in the psilocybin gene cluster. But really, I'm just interested in that population question. And that culture collection that I'm starting will hopefully be a resource for anyone else who wants to innovate psilocybin in the future.
And could this then be a reason why we can attract you back to South Africa?
Oh, you can attract me back there anytime, just with the promise of a spatlo. Delicious.
Oh, what?
Ah, you know those buns filled with chips? Oh, cut me a piece.
Bunny chop. Yes. Okay.
Oh, yeah, okay, some of that too. Oh, it's what I dream of every night.
So the colleagues here, who I guess you guys could have also put something in the chat, but maybe Tuan has put up his hand to say that he'd like to ask a question.
Hi, Alistair. Thank you for the talk. Since I'm here, so I got the privilege of speaking, not chatting. So I have one question and maybe one comment. So let's start with the comment first about horizontal gene transfer on close related species. should we not thinking about not using parsimony to decide whether the gene is horizontal transfer, but actually looking at synteny, because if you are acquired, you're not likely to be at the same location. Although I don't know how to automate that at the mass scale.
Yeah, I don't know how to show convincingly that it's a gene gain over a gene loss. Yeah.
Because if the gene that have different location on the thyroid group, it's not likely to be gained. I mean, of course, there's rearrangement, but if you look at the immediate neighboring of it, it probably, so synteny should be one of the way, but automate synteny in mass scale, I don't know how we're going to do that. So
how good are those assemblies?
Some are pretty good. So, okay, and then on to my question about RNAi, that's fascinating work, by the way. So should we not thinking about if we use conserved housekeeping gene as a target, would we be risking targeting beneficial fungi for the plants?
Yeah, well, that's kind of why I used the molecular barcoding genes. So you'd have to say that the molecular barcoding genes aren't satisfactory to identify species. If you want to go down that line, the reality is with rust fungi, it's pretty safe because all rusts are plant pathogens. So with rusts, it's kind of an open and shut case. With other fungi, then it's probably a little bit trickier because let's say you were targeting, you probably wouldn't target LSU, but let's say you were targeting ITS and you'd synthesize double-stranded RNA across the 5.8S, then even when it's fragmented, you're going to have sections of that of 20 to 24 base pairs that hit something, right? So whether it's enough to knock down the gene in beneficial fungi is a question that I've got no idea of the answer. Jojo's got a question. Josephine, she says that only one spore type do not take up double-stranded RNA. Do you know why that is? Jojo, I wish I knew. But that spore type that doesn't take it up is germinating to not infect. It's germinating to put out a basidiospore. So it could have something to do with that. It would be so cool to know whether basidiospores take it up when they germinate, because then they infect. So yeah, can't stop meiosis, but we can stop infection.